Function and characteristics of the myocardium. The heart is a muscular organ about the size of a closed fist that functions as the body's circulatory pump. Cardiac muscle, also called heart muscle or myocardium, is one of three types of vertebrate muscle tissues, with the other two being skeletal muscle and smooth muscle. It is an involuntary, striated muscle that constitutes the main tissue of the wall of the heart. The cardiac muscle, myocardium, forms a thick middle layer between the outer layer of the heart wall, the pericardium, and the inner layer, the endocardium, with blood supplied via the coronary circulation. It is composed of individual cardiac muscle cells joined by intercalated discs, and encased by collagen fibers and other substances that form the extracellular matrix. Currently, a large number of methods are used in medicine to investigate both myocardial morphology and function. In English language literature, cardiomyocytes are generally categorized into two major types of cardiac muscle cells, myocardial contractile cells and myocardial conducting cells, or pacemaker cells and force-producing cardiomyocytes of the ventricles and atria. Myocardial contractile cells make up the bulk, 99%, of the cells in the atria and ventricles. The contractile cells conduct impulses and are responsible for the contractions that pump blood around the body. Myocardial conducting cells, 1% of cells, make up the conducting system of the heart. With the exception of Purkinje cells, they are usually much smaller than the contractile cells and have few myofibrils or filaments necessary for contraction. They are similar in function to neurons in many ways, although they are specialized muscle cells. Myocardial conducting cells initiate and propagate an action potential, electrical impulse, that travels throughout the heart and causes contractions that propel the blood. In Russian language literature, cardiomyocytes are usually subdivided into three main types of cardiac muscle cells. Typical, working, cardiomyocytes, predominant type, function, contraction. Atypical cardiomyocytes, less myofibrils, function, generation and conduction of impulse. Endocrine, secretory, cardiomyocytes secreting atrial natriuretic peptide, ANP. Electron micrograph of ventricular, left, and atrial myocyte, right, showing location of ANP storage granules in a mouse model. Captured by Dr. Stephen C. Peng from Queen's University. Atrial natriuretic peptide, ANP, or atrial natriuretic factor, ANF, is a natriuretic peptide hormone secreted from the cardiac atria that in humans is encoded by the NPPA gene. Natriuretic peptides, ANP, BNP, and CNP, are a family of hormone/paracrine factors that are structurally related. The main function of ANP is causing a reduction in expanded extracellular fluid, ECF, volume by increasing renal sodium excretion. ANP is synthesized and secreted by cardiac muscle cells in the walls of the atria and the heart. These cells contain volume receptors which respond to increased stretching of the atrial wall due to increased atrial blood volume. What is the difference between a true syncytium and a functional syncytium? The syncytium is a cell-like structure developed by joining two or more cells together. The key difference between true syncytium and functional syncytium is that true syncytium is the skeletal muscle cell that is multinucleated. The functional syncytium is the cardiac muscle cell that is not long and uninucleated. Skeletal muscles and placenta are two important true syncytia. Skeletal muscles provide rapid communication and response between the muscles and the brain, and the placenta acts as a barrier between the mother and foreign cells. The functional syncytium is the contractions that allow the heart to work as a unit. These begin with pacemaker cells, and they are self-excitable. There is terminological confusion in the literature regarding the terms syncytium and symplast. We believe that symplast is a multinuclear structure formed as a result of the fusion of cells of the same type. That is, it is one multinucleated cell. Examples of symplasts: striated muscle fiber of skeletal muscles, osteoclast, giant cell of a foreign body. Synonyms are essentially terms such as true syncytium, anatomical syncytium. We do not use these terms. A syncytium is a structure consisting of autonomous cells connected by cytoplasmic bridges. Examples of syncytium: myocardium, a bundle of unitary, visceral, muscle cells, embryonic connective tissue, mesenchyme. In humans, the precursors of germ cells develop in the form of syncytium, oogonia in female embryos and spermatogenic cells in mature men.